Good day, this is Professor Will, CBMD PhD. Today is Friday, January 10, 2020. It is 10:44 and 12 seconds p.m. Eastern Standard Time. Today's reading, Transcriptional Regulation of Prokaryotes. So let's get started here. The first step in gene expression is the transcription of the coding DNA sequences to discrete RNA molecules. Specific DNA regions defined as promoters are recognized by the transcribing enzyme, the DNA-dependent RNA polymerase. The RNA polymerase binds to the promoter and initiates the synthesis of the RNA transcript. The enzyme catalyzes the sequential addition of the ribonucleic to the growing RNA chain in a template-dependent manner until it comes to a termination signal terminator. The DNA sequence between the start point and the termination point defines a transcription unit. An RNA transcript can include one gene or more. Its sequence is identical to one strand of DNA, the coding strand, and complementary to the other, which provides a template. The base at the start point is defined as +1, and the one before that is -1. Positive numbers are increased, going downstream into the transcribed region, whereas negative numbers increase going upstream. The immediate product of transcription, termed primary transcript, which extends from the promoter to the terminator, is almost always unstable. In prokaryotes, messenger RNA, mRNA, is usually translated concomitantly with being transcribed. It is rapidly degraded when not protected by the ribosomes. Whereas ribosomal RNA, rRNA, and transfer RNA, tRNA, are cleaved to give mature products. Transcription is the principal step at which gene expression is controlled. DNA signals and regulatory proteins determine whether the polymerase will choose to transcribe a certain gene and whether the whole process of transcription will be accomplished successfully. The timing of transcription of specific genes is influenced by environmental conditions and by the growth cycle phase. A molecular picture of how genes are transcribed. The nature of the regulatory mechanisms that control transcription are far from being complete, but lots of progress has been made. Work on relatively simple organisms, bacteriophages and bacteria, has provided new insights into the mechanisms that are involved in the regulation of gene expression, transcriptional control mechanisms being among them. Although there are significant differences in the organization of individual genes, and in the details and the complexity of the regulatory mechanisms among prokaryotes and eukaryotes, it is clear that the basic principles are shared among all organisms. Due to the relative simplicity of the prokaryotic biochemical pathways, their easy manipulation in the laboratory, and the advanced tools that are available for changing their genotype and for testing the resulting phenotype, it is easier to infer these basic principles by studying prokaryotes. RNA synthesis is catalyzed by the enzyme RNA polymerase. The reaction involves the incorporation of ribonucleoside 5-transpose triphosphate precursors into an oligoribonucleotide transcript based on their complementarity to bases on a DNA template. RNA polymerase catalyzes the formation of phosphodiester bonds between the ribonucleotides. The formation of phosphodiester bonds involves a hydrophilic attack by the 3-transpose -OH group of the last ribonucleotide in the chain on 5-transpose triphosphate of the incoming ribonucleotide. The incoming ribonucleotide loses its terminal 2-phosphate groups, which are released in the form of pyrophosphate. In this manner, the RNA chain is synthesized from the 5-transpose end toward the 3-transpose end. The best characterized RNA polymerases are those of eubacteria, for which E. coli is a prototype, unlike eukaryotic cells in which various types of polymerases are dedicated to the synthesis of the certain types of RNA. In eubacteria, a single type of polymerases appear to be responsible for the synthesis of mRNA, rRNA, and tRNA. The dimensions of bacterial RNA polymerase are approximately 90 times 95 times 160. The molecular weight of the complete E. coli enzyme is approximately 465 kDa. About 7,000 molecules of RNA polymerase are present in E. coli. But the number of molecules engaged in transcription at any given time varies from 2,000 to 5,000 depending on the growth conditions. The DNA sequence that is being transcribed by RNA polymerase is transiently separated into its single strands, with one of the strands serving as a template for the synthesis of the RNA strand. This region is therefore defined as the transcription bubble. As the RNA polymerase moves along the DNA, it unwinds the duplex at the front of the bubble and rewinds the DNA at the back, so that the duplex behind the transcription bubble reforms. Thus, the bubble moves with the RNA polymerase and the RNA chain is elongated. The length of the transcription bubble varies from 12 to 20 BP. The length of the transient hybrid between the DNA and the newly synthesized RNA sequence within the transcription bubble is a matter of controversy, and the estimates range from 2 to 12 NT. Beyond the growing point, the newly synthesized RNA chain enters a highly affinity binding site within the RNA polymerase. The RNA polymerases of certain phases consist of single polypeptide chains. These polymerases recognize a very limited number of promoters, and they lack the ability to change the set of promoters from which they initiate transcription. In contrast, bacterial RNA polymerases consist of several subunits. The most studied bacterial RNA polymerase, the one in E. coli, exists in two forms of holoenzyme and a core enzyme. The holoenzyme is capable of selective initiation at promoter regions, whereas the core RNA polymerase is capable of elongation and termination, but not selective initiation. The two forms of the polymerase consist of two identical alpha subunits, one beta subunit, and one beta transpose subunit, but only the holoenzyme contains an additional subunit, one of several sigma proteins. The alpha subunit plays an important role in RNA polymerase assembly. The alpha subunit plays some role in promoter recognition and the interaction of RNA polymerase with transcriptional activators. The beta and beta transpose subunits together constitute the catalytic center of RNA polymerase. The beta subunit was demonstrated to contact the template DNA, the newly synthesized RNA, and the substrate ribonucleotides. The beta transpose subunits contacts the RNA chain as well. Mutations in the genes that encode B and beta transpose show that both subunits are involved in all stages of transcription. The sequences of beta and beta transpose show homology to the sequences of the largest subunits of eukaryotic RNA polymerases. This conservation through the evolution of hints that the mechanisms by which all RNA polymerases catalyze transcription share common features. The assignment of individual functions to the different subunits of the core polymerase are only a rough estimation because most likely each subunit contributes to the activity of the enzyme as a whole. The sigma factor is involved only in transcriptional initiation. Its function is to ensure that the polymerase binds in a stable manner to DNA only at promoters, not at other sites. There are several types of sigma factors in the bacterial cell. The major sigma factor in E. coli, which is required for most transcription reactions, is sigma exponent 70. Although sigma 70 has domains that recognize the promoter DNA sequence as an independent protein, it does not bind to DNA, perhaps because its DNA binding domain is sequestered by another domain of the sigma 70 molecule. When sigma exponent 70 is shortened from the N-terminus, it is able to bind to DNA, suggesting that the N-terminal region has an inhibitory effect on the ability of sigma 70 to bind to DNA. However, upon binding to the core enzyme and the formation of the holoenzyme complex, sigma exponent 70 undergoes a conformational change, and it now contacts the region upstream of the start point. The dogma is that the sigma factor discharges from the core enzyme when the abortive initiation is concluded and RNA synthesis is successfully initiated, although findings suggest that, at least in some cases, sigma can remain associated with the polymerase at post-initiation steps. The released sigma factor becomes immediately available for use by another core enzyme. E. coli cells contain about 3,000 molecules of sigma to the exponent 70, enough to bind about 1/3 of the intracellular core RNA polymerase. Bacteria polymerases have to recognize a wide range of promoters and to transcribe different genes on different occasions. Specificity of gene expression is in part modulated by substituting one species of sigma for another, each specific for a different class of promoter. In Bacillus subtilis, sigma factors are implicated in the temporal regulation of sporulation. In E. coli, alternative sigma factors are used to respond to general environmental changes. The sigma factors are named either by their molecular weight, e.g. sigma exponent 70, or after their genes, which are usually termed RPO, e.g. RPOD for sigma exponent 70. When cells are shifted from low to high temperature, the synthesis of a small number of proteins, the heat shock proteins, transiently increases. The sigma exponent 32 protein, RPOH, is responsible for the transcription of the heat shock genes. The basic signal that induces the production of Sigma exponent 32 is the accumulation of unfolded proteins that results from the increase in temperature. The heat shock proteins play a role in protecting the cell against environmental stress. Several of them act like chaperones, preventing the unfolding denaturation of proteins. The heat shock proteins are synthesized in response to other conditions of stress, implying that the production of sigma exponent 32 is induced by conditions other than elevation in temperature. The sigma exponent 32 protein is unable and it is rapidly degraded when it is not needed. Another sigma factor, sigma exponent E, appears to respond to more extreme temperature shifts that lead to the accumulation of unfolded proteins, which are usually found in the periplasmic space or outer membrane. Less is known about this sigma factor and about the genes it controls. When E. coli and other enteric bacteria are nitrogen limited, the synthesis of a number of proteins is dramatically induced. The increased production of these proteins enlarges the capacity of cells to produce nitrogen-containing compounds and to use nitrogen sources other than ammonia. The expression of the flagellar genes under normal conditions depends on sigma exponent 28, also known as sigma F. When E. coli cells are starved, they shift from the exponential growth phase to the stationary phase. The ability of the cell to cope with starvation depends on the production of many proteins. This is enabled due to the synthesis of sigma S, RPOS, which transcribe the relevant gene. Unlike E. coli, when B. subtilis cells are starved, they can form spores. Sporulation involves differentiation of the vegetative bacterium into a mother cell, and a forespore of the mother cell lyses and a spore is released. The process of sporulation involves a drastic change in the biosynthetic activities of the bacterium, in which many genes are involved. This complex process is concerted at the level of transcription. The principle is that in each compartment, the mother cell and the forespore, the existing sigma factor is successfully displaced by a new sigma factor that causes the transcription of a new set of genes. Communication between the compartments occur in order to coordinate the timing of the changes in the mother cell and the forespore. The promoters identified by the various sigma factors are organized similarly, having their important elements centered around 10 and 35 NT upstream from the start point, except for sigma 54, whose promoters have slightly different characteristics. On another topic, how does RNA polymerase find the promoter sequences? How does it identify a stretch of 60 BP which defines a promoter in the context of 4 times 10 to the power of 6 BP that make up the E. coli genome? The first model assumes that RNA polymerase moves in the cell by random diffusion. It associates and disassociates from loose binding sites on the DNA until, by chance, it encounters a promoter sequence that allows tight binding to occur. According to this model, movement of an RNA polymerase molecule from one site to the DNA to another is limited by the speed of diffusion through the medium. However, this parameter might be too low to account for the rate at which RNA polymerase finds promoter. The second model addresses this problem by assuming that once an RNA polymerase molecule binds to a DNA sequence, the bound sequence is directly displaced by another sequence. The enzyme exchanges sequences very rapidly until it binds to a promoter that allows an open complex to form and transcription initiation to occur. According to this model, the association of the polymerase with DNA sequences and their dissociation are essentially simultaneous. Thus, the time spent on site exchange is minimal and the search process is much faster in comparison to the speed calculated based on the first model. This model fits the accepted notion that core polymerases that are not busy in transcription are stored by binding a loose site on the DNA. The third model assumes that RNA polymerase binds to a random site on the DNA and starts sliding along the DNA molecule until it encounters a promoter. Good day, this is Professor Will, CBMD PhD. Today, Saturday, January 11, 2020. It is 1:06 a.m. Eastern Standard Time. And this concludes my reading on transcriptional regulation in prokaryotes. Hope you enjoyed listening. Have a great day.